So, I'm Bapst. This is some work that I've been doing as a postdoc in Sandy Carlson's lab at UC Davis. I'm not actually at UC Davis, this is the question I keep getting asked. I'm actually a remote postdoc, and I tend to live in the Black Hills, and I'm actually also an adjunct at South Dakota School of Mines in the Black Hills. So, this is work that we've been doing, looking at morphology. People keep saying at this conference that phylogenetic paleobiology is apparently important. If so, then we really need to care about morphology, because morphology is the key to generally doing systematics in paleontology. Of course, that assumes that shared morphology is actually going to reflect shared evolutionary history. And just really how reliable is that assumption? That relationship probably varies among different groups, particularly given the sort of quality of data that we have for different fossil groups. Different fossil groups are very, very different. And how can we know when it's wrong? How can we know when morphology might actually be misleading us? We want to know when that might actually be occurring, and one way to do that is that we can compare morphology to an independent data set of molecular data for some set of extant taxa. So what's some fossil-rich group where we would want to know about morphology and has some extant diversity? Well, the Rhynchonellids, a group of articulate brachiopods, have 19 living genera, but back in the deep time, they had more than 500 extinct genera. So a very diverse group. We'd love to be able to reconstruct the relationships. How good is their morphology for doing that? So the Rhynchonellids, as I said, they're articulated brachiopods. There's a different group of brachiopods called inarticulates. That's basically the two major groups of brachiopods. They have these spiralophore lobivores that they use to filter feed. If you don't know what a brachiopod is, it looks kind of like a bivalve, but it isn't. And they're not related at all. They're of their own family. There are about 10 out of the 19 of the extant genera live at bathyal or abyssal depths, so they're really difficult to collect. Thankfully, there has been molecular work done on them and morphological work too. In fact, in 2013, 2 studies came out, one using the morphology, one using the molecular, and as you can see, here's 2 trees. Always in this talk, I always mark off where the crown Rhynchonellids are with green. Now, it's very hard for you to see how these relationships compare to each other, so let's do a tangogram. Boom! You can see they don't really relate each other to at all, and there's a whole bunch of unshared taxa. What are those? Well, those are their outgroups. So the morphology study used a bunch of Paleozoic, supposedly stem Rhynchonellids as the outgroup, and the molecular study used a bunch of non-Rhynchonellid outgroups, including a number of inarticulate brachiopods. Okay. If we look at how traditional taxonomy falls out, each of these colors is a different superfamily of Rhynchonellid brachiopods. Doesn't look very good for the traditional taxonomy. Anyway, so the molecular and the morphological topologies don't even compare very well to each other for the extant Rhynchonellids. What's going on? What can we do in order to get at this further? Well, we want to account for these differences in taxonomic coverage. We want to include as many crown Rhynchonellids as we could, say, in the morphological data. Try to also code out some of the characters for the non-Rhynchonellid outgroups. And so that's actually what we did. We revised our morphological data in the Carlson lab, and we added 10 more characters. And also, what about just combining the 2 data sets, taking the morphological data, taking the molecular data, and seeing what sort of story they tell together? Maybe it will support one topology or the other, or maybe it will find some third alternative topology. So we did phylogenetic analyses using maximum parsimony and PAUP. All of those were unweighted or equally weighted, either way you want to say it. We did Bayesian analyses with Mr. Bayes. We used the same model sequence change as the original molecular study used. And we used 2 different sort of configurations for model settings and priors for the morphology, where we either relaxed a lot of the assumptions about how the morphological data is evolving, and we also did a model with very strict assumptions in order to sort of maximize the information content that might be in the data. So we did both combined analyses. We also did a number of analyses with just the new morphological matrix, and including one reanalysis of the molecular data, just to see if anything changed. And we also did several with different variants of sets of taxa. One where we had all of the taxa, which everything that we had the morphology coded for, and also where we just had those taxa we had both molecular and morphology for. Okay, well, that adds up to about 12 analyses, and then not to mention having to compare it also to the original topologies, it gets really messy. How do we make sense of all these analyses against each other? Well, I was looking around for some way of doing this, and so finally I just invented something. Probably someone has invented this before me, but I put it up on a blog post, and so far 300 people have looked at it. No one's told me that someone's invented it before, so I don't know. Anyway, just comparing summary trees, drop all the taxa that aren't shared between two topologies, and then basically just count how many splits between the two trees aren't in agreement with each other. Basically, a lot of these consensus trees that I was getting out of the analyses were poorly resolved, and Robinson folds, so it's going to penalize the distance between those two trees because it has polytomies. I really want to know just how much the two trees actually argue against each other, how much they contradict. And so I came up with this metric, just count the number of splits that are contradicted by some split in the other tree, add those together, you divide by the total number of splits that could actually disagree. And so, for example, the contradiction distance between a star tree and a fully resolved tree is 0 because, of course, the star tree isn't inferring anything about the relationship, so how could it argue? And two completely resolved topologies that are completely contradictory, the contradiction distance will be 1. Okay, so that's a bunch of pairwise distances, and that's a scary table, oh my god. But anyway, one thing you should see immediately is that the original studies, the contradiction distance between them is 1. They're completely disagreeing with each other. Okay, but here's some takeaways you can take once you stare at that table long enough. One is that all of the morphology-only analyses really disagree with each other. They all infer very different topologies. However, they all agree with the Bayesian topology, which was relatively not well resolved, but had some resolution within the crown Rhynchonellids. So that was sort of a poorly resolved compromise of morphology analyses. And so you can see this here. So here's this poorly resolved compromise. You can see there's a big polytomy there. And here's the original molecular tree. And you can see that the doesn't have a whole lot of congruence. And furthermore, you can see that it's not even really resolving monophyletic Rhynchonellids. The pterobrachylids, which are another group of inarticulates and were used as an outgroup in the molecular study, are actually coming out in this giant polytomy that contains a whole bunch of morphological taxa. So the morphological data doesn't even have the ability to pull out the pterobrachylids from the Rhynchonellids. Okay, now the combined analyses, well, the Bayesian combined analyses and all the molecular analyses, including our reanalysis, had very little disagreement among them. And so here's our reanalysis of the molecular data. There was a little bit of shifting around of the relationships when we reanalyzed it, even though we reanalyzed it supposedly exactly the same way they did. And there's very good congruence with combined analysis, which there's only one little change here where the cryptopora and Notoceria, these two taxa flipped around. So the morphological data did impact the molecular data a little bit, but not very much. The molecular topology is winning out. And if you compare that to that poorly resolved morphological tree, oh, there's not very much congruence, and there's that contradiction distance that's pretty big. So the last one was 0.06. The one between the morphology and the combined analysis, pretty bad, 0.31. Okay. The maximum parsimony combined analyses, the maximum parsimony combined analyses, which differed in the number of taxa, they each produced their own unique topologies that were actually very different, and they were also very different from any other analysis. The morphology-only analyses, the Bayesian combined analyses, the maximum parsimony combined analyses all all looked very weird too. Lots of ladder-like pectinate plates, ladder-like pectinate plates. But anyway, so the molecular data is winning out in the Bayesian combined analyses. But how much is that just due to having 53 times more molecular characters than the morphological data? So what about some sensitivity analyses? Well, we can, if we just sort of take the morphological data, we can repeat it, say, 53 times, and then we've got two partitions that are the same size. Hey, what happens if we run that? It won't converge in Mr. Bayes. Okay, let's try something else. So, let's take some morphological tree, in this case, one of the morphology-only parsimony analyses, and we're going to simulate on it binary characters that are parsimony informative and perfectly support that tree. We're going to simulate a partition that's the same size as our original morphological partition. We're going to put it together with the molecular data, and we're going to run it as a combined analysis. What happens? If that's going to tell us basically what happens if our morphological data had been perfect, it had been the best signal we could have imagined. The one that wins is the Bayesian combined analysis, which looks just like a molecular topology. The only thing that's changed is, Cryptophora has moved around again. It's flipped from one being nested in one clade to being nested in another. And so that sort of results in some increase of the contradiction distance. Okay, so that Cryptophora taxon, that's really moving around a lot. What's going on there? Well, remember, it's 18S and 28S ribosomal DNA? Well, it turns out that actually a number of the taxa, remember they're hard to sample. Some of these deep dwelling brachiopods, they only got one of those for them in terms of the ribosomal DNA, and Cryptophora, it turns out, also has a deletion inside the one they did get, and so it's missing 60% of its molecular data, and so Cryptophora and Acanthobasiola, they end up flipping around a lot between the different analyses, and they're also missing the most molecular data. The ones that are missing all the morphological data over here are the inarticulates. The inarticulates, they always come out in the same place that the outgroup, right? And so a lot of the characters for Rhynchonellida don't apply to them. But the taxa that are flipping around are our combined analyses and our sensitivity analyses. They are missing a lot of their molecular data. And so, yes, the morphology is impacting things, but they're impacting things only for the taxa that are missing, 60% of their molecular data. Okay, so, just to sum up, the morphology only analyses could, generally contradicted each other or weren't very well resolved. The molecular only and the Bayesian combined analyses were fairly congruent with each other, and that can be seen with the contradiction distance that I came up with. The sensitivity analyses suggest that the large size of the molecular portion may be a controlling factor, because even if we had perfect 66 morphological characters, still wouldn't have gotten the morphology only tree or any sort of moving away from that molecular topology. And still, the small number of morphological characters did impact that much larger molecular analysis than the combined analyses, but it was only for those taxa that were missing a large number of their molecular data. Really, the morphological data seems to be pretty noisy. That seems to be what's coming out of this, but is that due to sort of long branches? The crown of the Rhynchonellids could go all the way back to the early Paleozoic. So really what we needed to do, and that's our next direction forward, is to start coding a bunch of fossil taxa within the Rhynchonellids. And within the Rhynchonellid crown, what we think is the Rhynchonellid crown, not the stem, in order to actually test whether or not there is actually some sort of pattern in morphology that's going on. So I'd like to thank NSF for funding, and also April Wright and Peter Smith for discussing various things with me. And I'll open it up for questions.